I'm the Chief Business Officer at Valo Therapeutics. And Valo, to cut to the chase, we'll do it in one slide. So Valo is developing an immunotherapeutic platform in cancer. It's based upon the use of oncolytic viruses. In the first iteration, it's using oncolytic adenoviruses as adjuvants to deliver very potent peptide vaccination immune responses in oncology in virtually any solid tumour. So that's the background. The company, so I kind of restate it, we feel like we're reinventing cancer immunotherapeutics using oncolytic viruses. In the first iteration, PeptiCRAD, we're using oncolytic adenoviruses, and I'll tell you a lot more about PeptiCRAD. And we probably won't have enough time to talk about PeptiENV, which is a follow-on technology where we're doing a very similar thing with enveloped viruses such as HSV, Vaccinia, and VSV. The company span out of the University of Helsinki at the very end of 2016, and to be honest with Valo, in Finnish, just as a bit of education, means light. And so what the company is doing is shining the light on tumours to make them more visible to the immune system. I was at the company 6 months before I worked that out. So the team is split geographically. So all of the science is done in Helsinki. And operationally, we're based in Oxford, just down the road. So the CEO is Mike Steen, and Mike is a kind of serial healthcare entrepreneur who most recently was responsible for the biggest spin-out fundraising in the UK biotech history when he spun out Oxstem from the University of Oxford and raised £16 million. I'm Scott Cutler and I'm the Chief Business Officer. My background is I have a PhD. I worked at Roche initially, went into business development about 13 years ago at small and bigger companies including Chroma Therapeutics and Ipsen, and I'm now the CBO at Valo. The team in Helsinki is highly experienced. I won't go through them all, but they're highly experienced. Every one of them was part of a company called Oncos, which was a pioneering company in the space. It took the first oncolytic adenoviruses into patients, and when that company was bought out by Targovax in Norway, who are now in phase 2 with that virus, it was this team that developed that virus. So there's a rich heritage of development of oncolytic adenoviruses within Valo. So what does the therapy do? I've been through some of this already, but it's an immunotherapeutic platform, very flexible, very adaptable to any solid tumour type. Importantly, it's synergistic with checkpoint inhibition. And we all know that checkpoint inhibitors, if you're fortunate enough to respond to, you're one of the 15% of patients that responds. That's fantastic. But there's a need to find other synergistic mechanisms to open up the immune system to make the tumour visible to the immune system, that the checkpoint inhibitors can then come in and block the blockade. We can apply this technology to any existing cancer peptide vaccine strategy, and that will become obvious in the next couple of slides. And we can also apply the technology to other oncolytic adenoviruses. We've chosen in developing our own virus, but we can work with other third-party viruses. In fact, we've just signed a collaboration that I'll tell you a little bit about. The oncolytic viruses themselves work. There's a cartoon here to show you kind of what goes on. So, especially in the adenovirus field, the viruses are engineered so that they'll only infect and only replicate and cause lysis within tumour tissues, tumour cells. And often they're engineered with, they're armed with various transgenes that stimulate the immune system, for example. And I'll describe our virus in a second. So there are two kind of overarching mechanisms that these viruses work by. One is the oncolysis that's caused as the virus replicates, and as it replicates and kills cells by oncolysis, the neoantigens and death signals are exposed to the immune system, to the antigen-presenting cells, and if you're fortunate enough to do that in the right way, you can get a robust systemic anti-tumour T-cell response. The problem is that most of the immune response, the CD8 positive responses, are to the virus and not to the tumour. And what our technology does is reverse that. So how does it work? So if you look on the left-hand side, it represents a generic adenovirus. We know that the capsids of those viruses are negatively charged. And so the idea became, well, could we add peptide antigens to the outside of the virus? And so what we did was we attached lysine tails to the amino terminus of any peptide, and you can decorate them by electrostatic interactions. The peptides stick and they form peptidograd complexes. Those complexes, depending on the peptide, can be thousands of peptides for every virus particle. And so what that offers us is the ability to put different types of peptides onto these viruses which act as adjuvants. And so we can use well-established peptide vaccines, and I'll show you some data using MAGE-A3 and NY-ESO-1. We could put newly identified peptide antigens that are involved in tumours, or the promise at the end of the day will be to deliver neoantigens so that when you go to the doctor you get a tumour biopsy and it's deep sequenced. The technologies are advancing rapidly to be able to tell you what the most important neoantigens are in that individual patient's tumour. Once we know what those are, we can make the peptides, put them on the outside of the virus. It's the same virus that we would always use, so there's no need to re-engineer a different virus for each individual patient. So I think that's the promise of the technology at the end of the day. I've said this already, so we're taking peptide vaccines which are not very immunogenic, making them much more immunogenic, and directing them to CD8 positive T cell responses to the peptides that you put on the outside of the virus. Our virus is described on the left-hand side here. It's engineered in the same way as I said before, but we are expressing two transgenes, CD40 ligand and OX40 ligand, which stimulate both arms of the immune system, so the adaptive and the innate immune system. And so that in that lysing tumour, you've got a perfect storm. So you've got the antigen-presenting cells being professionalised, and that you're in the presence of a high concentration of the peptides on the outside of the virus. So here's one piece of data using a PBMC model where human PBMCs are put into mice that carry a melanoma on their flanks. And you can see that the bottom two lines are peptigrads that express NY-ESO-1 or MAGE-A3, which are common peptide vaccines which have been in many patients and shown to be immunogenic, just not enough in a wider population. And you can see that indeed you get a reduction in tumour growth, which is statistically significant. More interestingly, when you look and these mice are fully humanised, so their immune system has been ablated, replaced with a human immune system. And in those mice, there's a lot on this slide, but the important point is that if you look at mock animals, you can see that the y-axis is showing the percentage of cells, CD8 positive T cells in the blood that recognise the two antigens that we've put on the outside of the virus, so MAGE-A3 and NY-ESO-1. In mock animals, you don't see anything that recognise, there's no CD8 positive T cells there. In animals that are given peptides on their own, in poly-IC, you can see that you can get about 1% of CD8 positive T cells, which is a significant induction. You can see 1% of their cells maximum recognise, CD8 positive T cells recognise the antigens. But when you look ultimately at the far right hand side, you can see that 60% of peptigrad mice that have been given the virus with the antigens, have significant induction of CD8 positive T cells. The majority of them above 1%, but there are some animals that go up to as high as almost 30% of the CD8 positive T cells recognise the peptides on the outside of the virus. And I'm not an immunologist, but they tell me that's phenomenal results. So if you can recapitulate that in humans, that would be enormously opening up the window for checkpoint inhibitors, for example, and would be fantastic for patients. So I mentioned already that we have that we could use anyone's virus, and in fact we are developing our own virus that I showed you. We've recently just signed a collaboration with a company in San Diego called DNAtrix, and DNAtrix already have their own oncolytic adenovirus that's in phase 2 clinical trials in recurrent glioblastoma, and they're showing fantastic responses in those patients. When we met up with them, we immediately formed the idea that we should work together. This helps us enormously because it helps us, the companies are truly synergistic, but this gets us to validate our platform technology much more quickly in patients than we would if we had to develop our own virus which we're still doing, but it would take us longer to get into the clinic. We're sharing the development costs for that. But Valo retains the rights to its technology in all other indications with its own virus. And it gives us a presence for running clinical trials both in the US and in the EU, which is enormously helpful. I won't spend any time on this. We can do something very similar with envelope viruses. The only oncolytic virus that is licensed at the moment is T-VEC from Amgen, which is a herpes virus. So the idea is that we maybe would be able to boost the immune responsiveness of those type of viruses. I think we've said that, so I'd like to concentrate on our timelines. So the company spun out, as I said, at the very end of 20. We've raised our first real monies at the very end of 2016. We've raised about 9.5 million euros to date. We've just signed up to this collaboration with DNAtrix. Our lead candidate should be in the clinic using the DNAtrix virus at the end of this year, and the plan is then to go into probably a basket study of 4 indications in patients who express the MAGE-A3 and NY-ESO-1. And then as we move forward, we will decide what the indication should be for the full development. And we're continuing developing our own virus. In fact, that will be ready for going into CMC manufacture in April. So I've still got time. So what are we looking for? We're looking to raise, I said we've already raised 9.5 million. We have, some of that money was raised from Techis, which is now called Business Finland. And Business Finland is an innovation, the government Finland innovation body. And we have the possibility to reach through to another 3 million euros from that agency. 50% of the co-development of the cost for the trial with the DNAtrix virus have been covered by DNAtrix. So we'd like to raise around 10 million euros to help us complete all the studies we need to get into phase 1 and to run the phase 1b study. That will be a study with checkpoint inhibitors. If I didn't say that, I apologise. And we also want to continue with the development of our own virus. And lastly, we want to bring on and develop more of the PeptiENV platform, so the same technology, slightly different technology, but the same output with envelope viruses. Because there we could use a prime boost strategy with our PeptiCRADs, but also there's a much wider utility in melanoma and others if you think of T-VEC and some of the other oncolytic viruses, envelope viruses that are being developed. With that, I'd like to stop. I'm a little bit early, but if there's any questions, I'll be happy to answer them and share it.